Hello friends, welcome to BMH Learning. This video is about single-stranded DNA damage and repair. Single-strand break means one of the strands of double-stranded DNA molecule is damaged. It can be damaged in a number of ways, and similarly different mechanisms are used by cells to repair DNA damage. The diagram shows the direct single-stranded DNA damage. The damaged termini in the single strand is represented by red circles. DNA single-strand breaks arising directly from the disintegration of damaged sugars following DNA damage are rapidly detected by the sensor molecule poly-ADP ribose polymerase 1. Single-strand breaks also result from the action of repair enzymes that recognize and remove damaged DNA bases. A third type of single-strand break can also arise if topoisomerase 1 comes into close proximity to elongating replication forks, transcription bubbles, or DNA lesions. Despite arising in different manners, all single-strand breaks are repaired in overlapping repair pathways, collectively called single-strand break repair. And processing. Due to the presence of damaged DNA termini, the majority of single-strand breaks require processing. This was all. Thanks for watching.